Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Tarek Odali from Business Review, and I will be your host. It is our pleasure to have Piramal Pharma Solutions with us today, who will be discussing the strengths and challenges of analytical development in the pharmaceutical industry. Today's guest speaker is Dr. V. Srinivasan, Senior Group Leader, Analytical R&D at Piramal Pharma Solutions. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you do disconnect for any reason, please click on the link that you received via email to rejoin the session. In order to ask questions, you can send them in at any time via the questions widget. Just type them into the box at the top left-hand corner of your screen, then click Submit. We'll try to allocate some time at the end of the session to address any of the questions or thoughts that you've sent in. Please use the yellow Help widget if you require any technical assistance. You're also free to move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now, that having been said, please allow me to welcome today's speaker, Dr. V. Srinivasan.
Yeah, thank you, Tariq, for your warm welcome. Good morning to all, good afternoon, good evening, depending upon the time zone. I am Srinivasan from Piramal, PhD in chemistry, having 20 years of experience in analytical department. Presently, I am working for Piramal as a head analytical for Ennore, Chennai, India. Before jumping into the topic, let me take a minute to introduce about Piramal, what are the activities we are performing here. Piramal is a global contract research, development and manufacturing organization. We provide service across the whole drug development process, starting from discovery services, intermediate, API development and manufacturing, drug formulation, antibody drug conjugates, sterile fill, and finished products. Piramal has a global footprint across globally in US, UK, Canada, and in India and China. This is just basic thing about Piramal. The topic is what I am going to discuss here is the strength and challenges in analytical development pharmaceutical industry, which is very needy in these days. What is the challenge here? Like, until a few years ago, the quality of the drug substance was mostly determined by purity rather than impurities. A couple of years ago and all, normally people will see what is the purity of the compound, and but the thing has changed, and the quality of the drug substance was determined not only by the purity, but also the impurities present in the drug substance. The scenario is completely changed. Now we talk about what is the impurity level, not 5%, 0.1%, something like that. It originates because of regulatory bodies are more concerned on impurities, because some of the impurities are more potent than API, and they need to be quantified and controlled because of the health concern. This forces the regulatory bodies to enforce on tighter control on the impurity level. As I said in the earlier days, we concentrated only on purity. Now the thing has changed. Even few years ago also, if you are having a known impurities, means like you have to quantify anything above 0.15. Now it has come down even to a level of 0.1. People are talking about not 5% or 0.1%. It is really challenging because you should have a method to determine the things. So it is necessary to develop scientifically a sound method to determine the quality of the drug substance. How do we address these challenges? First is develop scientifically a robust method and validate the method and transfer the method to the end user quality control department. Though technology gives innovation products like UPLC or submicron columns and all, proportionately there is a demand for the end product also. As I said, sensitivity of the compound or percentage level of the impurity detection. Why analytical method development is required? It is required to establish the intent, identify purity and potency of the drug we use. Method development is required to support drug testing against a specification during manufacturing, quality release and stability studies. Why we require method? One, to support the manufacturing activity. The second is for the release of the compound, and third is for our stability studies. According to ICH, the most common type of analytical procedure is identification test, quantification test, quantification of the impurity, and limit test for the control of impurities. Method development is a continuous process. It will not stop. Once I develop a method, this is the final thing. It's not like that. It is a continuous process. The goal and purpose of the method should reflect the phase of drug development. I should know what type of development is required. Is it in phase 1, phase 2, or depending upon the requirement, our development also should go parallelly. When we talk about impurities, because this one is a purity, other one is a impurity. So what is this impurity? From where it comes? Impurities are unwanted chemicals which may have different therapeutic activity and may be potentially harmful. Therefore, they need to be identified, quantified and controlled during the development of API. This is a challenging one for the analytical chemist, how to identify the things and how to quantify the things. These two is the job of analytical chemist. Based on that, control will automatically come during the process. Impurities can be classified like organic impurities. This may be a starting material or impurities present in the starting material, your intermediate itself an impurity or byproduct or degradation product and the reagents. Inorganic impurities. This may be your catalyst or salt, buffer used in the process, heavy metals and sometimes a metal leaching from the equipment used. So whatever equipment you are using, from that also impurity may originate. The other one is residual solvents. This is the solvents used in the process and it has to be controlled as per ICH guideline. For example, if you are using a toluene, obviously toluene will contain a benzene, so there is a need to control benzene also and ethanol. So whatever solvents we are used in the process which cannot be completely removed as per the guideline, we should have a control on the solvent level. The most commonly used technique in any pharma industry is like HPLC and GC. This is a key tools used by the chemist to identify or quantify the analysis. HPLC methods like non-volatile compounds are analyzed by HPLC separation technique. Among that, reverse phase is the most commonly used separation mode. The other one is a GC method. If the compound is volatile, it can be directly injected into the system. If it is a non-volatile or it is a semi-volatile, it requires an extraction technique. Large volume of injection is a very useful tool here. Coming to HPLC, we talked about there are two phases. One is a normal phase, other one is a reverse phase. As a general guide, normal phase chromatography shall be used for separation of polar compounds and reverse phase chromatography for compounds that are moderately polar to non-polar range. Normal phase chromatography commonly involves the use of a silica column or a aminopropyl or cyanopropyl stationary phases. These columns may be used to separate polar compounds such as amines, anilines, phenols, and pesticides. Reverse phase chromatography is typically accomplished using a mobile phase mixture of water and another solvent, normally like acetonitrile or methanol is used. If the resolution is poor, gradient is used in combination of two or more solvents. The advantage of, as I mentioned, the technology is coming up like anything, and we are having a shorter column with a submicron particles and all. The advantage of using shorter column are back pressure, solvent saving, sensitivity will be very good, greater sensitivity, and reduced analysis time. All is achieved without compromising the resolution. Using these columns, gradient methods may be used to achieve very rapid analysis of samples that contain a wide polarity range of analyte. One example is like meborin hydrochloride, which is having 6 impurities. Still, I will say this is a challenge actually. Still, British Pharmacopoeia method of analysis is by TLC only. Still, it is following only a convention method. Thin layer chromatography is used for release of the compound. With the advancement and availability of column, we have developed a UPLC method using a submicron column, and all the 6 impurity has been separated with a good resolution, and the runtime is only 6 minutes. I will say challenge is you are able to separate all these 6 impurities within 6 minutes, and the strength is technology. As I said, UPLC and shorter column. The second example is donepezil hydrochloride. As per USP method, the runtime is 50 minutes. But we have developed a method which is only 8 minutes with the separation of all the impurities with respect to the API. I will say this is again, because of the shorter runtime, you are saving the solvent as well as the time and occupancy of the instrument. So our method, whatever thing we are developing during the development, we should focus not only the separation, also the runtime and long lasting for support to the manufacturer also. So during development, what are the criteria I have to look into that? One is method goal. If it is an early phase development, scientifically sound method is sufficient. The validation requirement is there, not required unless requested by a client. Documentation, good documentation practice should be there. If it is in phase 1 or phase 2, how? If it is in phase 1, method goal is partial validation, like a qualification is sufficient, and validation requirement, yes. If it is phase 2, it is required. Documentation wise, a written protocol and report is required for phase 2. If it is phase 3 plus, obviously, it should be a fully validated method with a protocol and report. So when we are working on development, we have to see in which phase the molecule is. Accordingly, we have to develop the things and validation requirement also. The basic criteria during separation goal is, or whenever you are developing a method, the separation goal should be like resolution should be greater than 2. If it is for a quantification, like when you are doing assay and all, our RSD should be more than 2%, and the pump pressure should be optimum. It should not be more also, and solvent consumption should be minimum. It means like your runtime should be short. So you have to plan accordingly. To save time as well as cost of analysis. The first one is mobile phase selection and optimization of mobile phase. The primary objective in selection and optimization of mobile phase is to achieve optimum separation of individual impurity along with the APA peak. The selection of mobile phase is always done in combination with selection of column. So it's like interlinked. We have to choose which one will be suitable. Following are the parameters which shall be taken into consideration while selecting and optimizing mobile phase. What are the things needs to be considered during a mobile phase preparation? One is buffer, if any, and its strength. What type of buffer we are choosing, and its concentration or strength has to be optimized. And pH of the buffer and mobile phase composition. Buffer and its strength play an important role in deciding the peak symmetry and separation. I'll take one by one. Yeah. You see the chromatogram. One is done with trifluoroacetic acid and mobile phase A. The other one is done with orthophosphoric acid. Both are done at 210 lambda. If you see the one which executed with TFA, baseline is not straight. It's like a step. It is coming down. So one disadvantage if the chromatogram is like this means we may not. One will be a sensitivity will be poor. Like we may not be estimating the correct amount of content present here because of the baseline. If you take the second one which is performed with orthophosphoric acid, the sensitivity will be good. Like the other one is 0.5% compound we injected, we are able to see a good sensitivity, whereas the same thing is not clearly seen or right quantity is not clearly achievable with respect to TFA. So when you are optimizing the mobile phase, all these criteria has to be keep it in mind before finalizing a method. The next one is the pH of the buffer and the pH of the mobile phase. pH plays an important role in achieving the chromatographic separations. It controls the elution property by controlling the ionization characteristics. Depending on the pKa, the drug molecule retention changes. Experiments shall be conducted using buffers having different pH to obtain required separation. One important thing we have to consider here is all columns will not withstand all pH. So it is important to maintain the pH of the mobile phase in the range of 2 to 8. Ideally, this is the ideal pH to maintain because all columns or almost all columns withstand this pH range. There are few columns available in market which can withstand above 8 also. So during the selection, we should be very careful that what type of molecule we are taking and what type of column needs to be used. If you see the chromatogram, first one is done with a pH of 3, the other second one is done with a pH of 3.2. Just a variation of 0.2 varied the retention time by 2 minutes. And one more caution has to be taken when you are adjusting the pH. Only the aqueous phase has to be adjusted for the pH, then you add your organic phase. That care should be taken whenever you are doing a pH adjustment. Next comes mobile phase composition. In reverse phase chromatography, the separation is mainly controlled by hydrophobic interaction between drug molecule and the alkyl chain of the column packing. Most widely used solvent in the reverse phase chromatography is methanol and acetonitrile. Commonly used solvents. I mean, if you take any method, mostly the thing will be like acetonitrile or methanol or combination of both. In few cases, even tetrahydrofuran is also used. Experiment shall be conducted with a mobile phase having buffers with a different pH and different organic phases to check for the best separation between the impurities. Here is a typical example. The first one is using a acetonitrile. If you see, you could see only two peaks. One is the main peak, the other one is the impurity. You see the second chart. It is done with a combination of acetonitrile and methanol, 1:1 ratio. You could see two peaks. Actually, it's not a single peak. One more peak is also inside that. This is achieved only by adding a methanol. So during the method development, we should be very careful when you are finalizing a method, because we should not miss out any impurities. Then comes the selection of column, like it's like an heart for any chromatographer. Yeah, because here you have a plenty or a variety of column selections. You can play around here. Following are the parameters of a chromatographic column, which are to be considered while choosing a column for separation of impurities and the degradants. One is length and diameter of the column, starting from 25 centimeter. Now it has come to 5 centimeter, also even less also. Its packing material, size of the particle, percentage of carbon loading, and encapping. Column plays a most important role in achieving the chromatographic separation. Most chromatographic separations are achieved due to wide variety of column available and due to flexibility to change the column and control each of the above parameter. Yeah, as I said, a lot of different dimensions columns are available in the market. It's really a good thing for the chromatographers to play around. Selection of column, as I said, commonly used columns for any method development is C18, C8, C3, phenyl, cyano, amino, and polystyrene. When we are going to work for a higher pH compound, a preferred column is polystyrene, which can withstand 1 to pH 13. Here is the polarity out. It starts like it from silica, cyano, amino, phenyl, C6, C8, C18. Normally C18 and C8 is used for any reverse phase method development, and silica is used for normal phase method development. This is another example citing the usage of two different columns. The first one is a X-bit shield RP18 column, where you could see the two peaks are little closer. Same thing when we have done with the Zorbax phenyl column, separation is good. It doesn't mean that Zorbax SB phenyl is superior and X-bit is not inferior. It's not like that. For this type of compound, separation is good in the phenyl. So when you are choosing a column, you have to choose such a way that the resolution between the two peaks is good. The next one is like a selection of detector. For like a column, we have the selection of detectors. The most commonly used detector for any method development is a PDA detector. Simultaneously, we can monitor and observe components at a different wavelength. And the next is a UV visible detector, which is a universal detector used for any HPLC instrument. But unlike a PDA detector, here we can see only 1 nanometer at a time. RI detector. The responses depend on the changes in refractive index of the eluting compound in the mobile phase here. But here normally glucose, fructose and all used are determined using RI detector. But one disadvantage I will say that here stabilization time takes more here compared to UV and all that. The next complementary detector is ALSD detector. It is used for detection of high molecular weight molecules. Here what we have to use like volatile buffers, and CAD is an ideal detector for non-chromophores, and gradient is also allowed in CAD detector. It is a very good complementary detector for non-chromophore compounds, and it supersedes RI or ALSD detectors. Fluorescence again it offers a greater sensitivity than a UV visible detector, and above this mass, as everybody knows, and commonly used in industries, a mass detector which is highly sensitive and selective. Next one is we have seen the column selection, we have seen the detector. The next is selection of solvent delivery system. Like chromatographic separations with a single element like isocratic. Here all the constituents of the mobile phase are mixed and pumped together as single element, which is a preferred one. Consistent, your pump pressure will maintain consistently, there will not be any abnormality. However, gradient elution is a powerful tool in achieving separation between closely eluting compounds or compounds having widely differing polarities. Very close elution things and all can be separated obviously by gradient system only. The important feature of gradient elution is that it is a powerful tool since the polarity and ionic strength of the mobile phase can be changed. We can alter the things. It's also important to optimize the program for initialization after each run. It's a very important thing here we need to point out here. When you are going or when you are using a gradient system, yes, you have to give initialization time. After the gradient, you should give equilibrium time so that carryovers can be avoided. The program for the initialization shall be optimized such that there shall be no carryover to the next run and the system stabilizes with initial combustion before next injection. It's a very important thing when you choose a gradient. A typical example is in the first chart, we could see an isomer which is very close to the main peak. It's not completely resolved. With the gradient, you are able to separate the isomer. The gradient plays a very important role, or a major close solution impurities can be separated by altering the gradient flow. Selection of flow rate. This will be very ideal for isocratic systems or even chiral analysis. Flow rate shall be based on the following data like retention time, column back pressure, separation of impurity, peak symmetry, theoretical plate. The preferred flow rate shall not exceed 2.5 ml per minute. So we should have a control, it should not exceed 2.5 ml per minute. A typical example is the first one is a chiral chart where we are unable to see the resolution. Same sample, same mobile phase. Only thing we reduce the flow with a 0.5 ml, we could able to see the good separation or resolution. Another important criteria is like column temperature. This also plays a major important role in achieving the symmetry. Always it is preferable to optimize the chromatographic condition with the column temperature as ambient. Normally it will be like 25 degrees preferred thing. However, if peak symmetry could not be achieved by any combination of column and mobile phase, then the column temperature above ambient can be adopted. Though we have the option to play with the column or mobile phase, still we have some problem in either separation or peak symmetry. Yeah, we can increase the temperature. The increase in temperature generally will result in reduction in peak symmetry and peak retention. Normally we can go from 30 degree to 60 degree. We can raise the temperature. The example here is like an interesting thing. Apart from temperature of the column, even the sample cooler also plays a role. If you see the example here, the sample cooler which is kept at 25 degree, that peak is not well resolved from the main peak, whereas the sample cooler kept at 15 degree, you are able to see a peak. It's well resolved from the main peak. So, like two things we have to consider. One is cooler temperature, sample cooler temperature, that also play a major role, and even a column temperature also, it also plays a major role in the symmetry of the peak. Then comes a selection of a detector wavelength. This is going to decide the fate of the compound, because we should work on the right wavelength, then only you will get the right picture. Selection of detector wavelength is a critical step in finalizing the analytical method for impurities and degradants, because we should be very careful here. Inject the impurity and API standard solution into the chromatographic system with a photodiode array detector and collect the spectra. Try to get as much information as possible. Also conduct force degradation studies and collect the UV spectra for all major degradation products. Overlay the spectra of all the compounds and select a wavelength which is most common and gives higher response for all compounds. So we cannot jump into conclusion that this is the lambda max for the compound immediately. Analyze the sample in the particular. Try to get much data from its impurities as well as degradation product. Overlay the things and arrive a conclusion. What lambda we need to use for this analysis. Selection of diluent for test preparation. Yeah, this also plays each and every parameter plays an important role in any method development. Diluent for the test preparation is selected initially based on solubility of the drug substance and known impurities. First, we have to check which solvent it is soluble and its impurities. However, finalization of diluent is based on the resolution of impurities, peak symmetry, and diluent blank injection interference. Like there should not be any interference because of the diluent. It has to be ensured that there is no interference. Analyte and impurity should not degrade in the selected diluent. And there should not be any degradation just because of your diluent. We should not see any degraded peak also. So for that, we have to inject the diluent blank and test the solution with the impurities and establish the non-interference of blank in the system. So before finalizing a diluent, we have to do the studies like any interference is there or any degradation is there. After the study only, we have to finalize the diluent. A typical example is, you see that first chart is methanol is used as a diluent, especially like acids and all. Yeah, if you add water, then only you will get a sharp peak or symmetrical peak. The second one is the diluent used here is methanol and water in the 1:2 ratio. You could see the difference between the first one and second one. Peak shape is very good here. Selection of test concentration and injection volume. Yeah, this is also another important criteria. The concentration should be nominal one. The test concentration generally chosen based upon the response of the API peak and impurities at the selected wavelength. You have to see all the parameters, like one is the concentration of the API, its impurities and the particular wavelength. So you have to optimize in such a way that you get a good response. The concentration should be lowered by increasing the injection volume, but it is to be ensured that the selected injection volume for the column is not overloaded. We should not overload the column when you are optimizing the concentration, but it can be lowered by increasing the injection volume. After the test concentration and diluent is finalized, the test solution has to be prepared and it has to be observed for any precipitation is formed. It's a very, very important thing. Normally, we will skip this, but it is very important because we may not be knowing what is exactly happening in the column. So it should not precipitate once it enters the column, which will mislead the results. So we have to observe, manually add this diluent and keep it at the bench top and observe that any turbidity is formed after 24 hours. Other technique in HPLC is a 2D technique, which is advanced thing, like it's also called hot cutting technique, which is ideal for analysis of impurities in the chiral pharmaceutical substance and simultaneously determination of enantiomeric excess. The first dimension, a reverse phase separation, is used to separate alkyl impurity from the API. The second API element is transferred to the chiral second dimension column for the determination of enantiomeric composition. It's called a hot cutting technique. Like when you inject the system initially, that you will see the achiral purity of the compound. Then the API is by diversion wall, it is sent to the chiral column where its chiral enantiomer is separated. In a single injection, you are able to see the. You can do both the analysis. It's like advancement in the HPLC technique. So far we talked about only on HPLC because most of the compounds are analyzed by HPLC. The other one, as I mentioned, like is GC. GC, like here the compound must be sufficiently volatile and thermally stable. And here the one limitation, few limitations are here is like inorganic and salts cannot be analyzed, limited sources to play the separation. Like in HPLC we have the option to play with the mobile phases and all, but here it's not, like a carrier gas is the only mobile phase. And here only left out option is the column. So we are bound only with the column selection. Based on the column selection, only we can change we can achieve the separation. Commonly used columns in the GC is HP1, HP5, DB17, DBVAC, and DB1701, DB624, which is a commonly used column for residual solvent analysis. And the recent development is a DBFFAP, which is a polyethylene glycol and diepoxide esterified with nitrothalic acid. This is very good column for especially when you are working with the acids and acetic acid, peak shape and all will be very good with this column. Commonly used detector in GC is a flame ionization detector, thermal conductivity detector, electron capture detector and the mass detector. Similar to HPLC, I have not touched on much about GC here. The principle or separation technique is as I explained in the HPLC. Same thing is applicable for GC also. But one thing I want to stress here, like one example is ethyl hydrocinamate. One compound was analyzed using a DB1 column where we are not able to see one impurity, whereas we are able to see with the RTX column, RTX 1701 column. The criticality is like this is very critical raw material for one of the API. Based upon the analysis with the DB1, that impurity is missed out, but we are able to see that impurity in the API. When we investigated, we found that the impurity is coming from the starting material itself. Again, to emphasize on this challenge and strength, it's on a typical example. Hydroxylamine, which is a genotoxic impurity, which was earlier determined by LCMS. LCMS challenge here is like it's not a easily available instrument in all the labs. One is its cost, another one is a trained person is required. We have just changed the determination by LC method using a derivatization technique. The same was validated and it is submitted to FDA also. Even FDA also approved the method. So here is the strength is alternate method. Challenge is again MS, considering its cost and the quality of the people. The second example is Veratrol, which is a key starting material in verapamil hydrochloride. I'll say this is again a challenge and strength I couldn't explain here. Veratrol is a key starting material in the verapamil hydrochloride API. If in Veratrol 2-methyl and 3-methyl are the impurities. If it is not determined in the initial stage in the raw material, it will carry over until the final API. One thing here is, initially we had a method and the source of Veratrol was from one vendor. The method was finalized based on the route of synthesis provided by the vendor. Accordingly, we developed a method. When we have moved to another vendor, his route of synthesis was different. We missed out to find out his route of synthesis impurities which yielded us or troubled us in the final compound. The message here is whenever you are developing a method, definitely even for the raw material also you should have a route of synthesis should be got from the vendor, and we should ensure that all the impurities is separated in the developed method. So far we covered about a major part of analytical method development. Once a method is developed, obviously it should go for validation. Once the analytical method is developed, it must be validated. As I said, the extent of validation evolves with the drug development phase. Need not be all methods to be validated. Depending upon the usage, it has to be validated. Validated method also gives unpredicted or unknown problem. Even though if the method is validated, as just now we discussed, the route of synthesis changed. Obviously, with the same validated method, we may not be in a position to achieve the desired result. So, validation is done with respect to predicted variable parameters. Whatever thing we initially assumed, with that only validation we are carrying out. The advantage of validation is, the biggest advantage of validation is that it builds a degree of confidence not only for the developer but also for the user. Although the validation exercise may appear costly and time consuming, it leads to better time management in the end. With better control on method validation, yes, we can achieve complete method, like obvious reasons, but method validation observe the shock of such conditions and pays more than invested on the process. It's Typical parameters recommended by FDA, USP and ICH for any validation is specificity, linearity, and range, precision, accuracy, solution stability, limit of detection, limit of quantification, and robustness. I will not touch more on that. This is already available in the guidelines. The last topic of our seminar is the method transfer. Once the method is developed, validated, and it needs to be transferred to the QC, which is a very important thing. During the transfer, because once it is transferred, then only the whole method is completed. Following are the points to be considered during a method transfer. The receiving laboratory will verify the equipment to perform the method testing is available. It is very, very important, though both the instruments are HPLC, there may be a different make, that also plays an important role. So we have to ensure that equipment or system is available, reference standard, methods are shipped from the sending laboratory to the receiving laboratory. Transferring laboratory shall provide necessary training to the receiving laboratory. We have to provide necessary training to the receiving laboratory. The receiving laboratory shall run through procedures, methods, and identify any issues prior to start of the execution of transfer protocol. Staff at the receiving laboratory shall be properly trained and qualified to run analysis method. We have to ensure that a proper training is given to the receiving laboratory person. Before that, a pre-approved protocol shall be reviewed, approved by both the laboratories, and shall follow appropriate procedures. Pre-transfer meeting should held to review the method transfer data. Before start of the activity, a meeting has to be arranged and all the things to be clarified, any outstanding issues, and to be remedies any deviations encoded of label. Whatever things is required has to be discussed in the meeting and sort out. After completing things, final test report shall be initiated to summarize the results obtained at a minimum. The final report shall outline the results obtained. It is highly recommended that method transfer process flow for each method is mapped. Once all the process flows are mapped, bottleneck and areas of concerns can be determined and mitigated. With a process in place and a process appropriately mapped, analytical method transfer has every chance of success. Here the message is like when everything is properly planned and it is properly transferred, there will not be any issue during execution of a method. One typical example here is like first one is a DMF from vendor 1, the second one is a DMF from vendor 2. If you see that in the first one, the impurity just eluted as a single peak. Actually, it is not single. It is 3 impurities are inside. Just only the brand only changed. So whenever you are doing any method validation or development and all, and when you are writing the transferring the things to the QC, you have to ensure that whatever reagent you make you have used, it should be reflected in the testing procedure. And the second example is like, as I said, instrument. We have to ensure that correct instrument is available at the sending as well as the receiving laboratory. Like Shimatsu, Vigilant or whatever instrument, we have to ensure that all the parameters are identical. If you see that example here, the first one is run in Shimatsu instrument and the second one is done in Vigilant instrument. Though both the instruments serve for the same purpose, the settings of both the instrument is slightly different, which evaluated or which resulted in not separation of the impurity in one instrument. So whenever we are doing a transfer, we have to ensure that identical condition is used in both the places. So far I covered from method development, validation and transfer. And I'll just take a minute to summarize what are the infrastructure, analytical infrastructure available at Piramal. We have HRMS, LCMS, GCMS, NMR, XRPD, UPLC, Rep HPLC, GC H-space, DSC, polarimeter, and other spectrometers. Piramal expertise in analytical sciences, what we do and where we can support like is analytical method development, analytical method validation, post-degradation studies, impurity isolation characterization, reference standard qualification, genotoxic content estimation by GC, HPLC, LCMS, stability studies as per ICH guideline. And thank you. And I hand over to Tariq for any question and answer. Thank you for your time.
Much. Just a reminder for the audience today, you can enter a question now. You can submit questions by typing them into the questions widget. So just go ahead and type them into the box at the top left-hand corner of your screen and then click submit. And we're going to try and get to a few of these, as many as we can before we run out of time today. So this one here is asking whether method validation is required if route of synthesis is changed by the vendor for the raw materials.
Yeah, answer is Tariq, obviously yes, we have to go for a revalidation because as I cited in my previous slide, example Veratrol case, once the route of synthesis is changed, obviously development and validation is required because whatever thing validation has been done is for predicted impurities only. Answer is yes, we have to go for revalidation.
Okay, lovely. Thank you for that. The next question right away is, how do I quantify assay for unstable compounds?
Right technique is NMR because wherever your compound is not stable, preparing a standard and qualifying the thing is very difficult. So in such cases, NMR is the ideal tool to go for quantification using a internal standard. Many standards are available like TCNB, malic acid, dimethyl fumarate, and depending upon the chemical shift, you can assign the compound or you can estimate the compound. The ideal technique will be NMR here.
Okay, thank you very much. And just a reminder for the audience, if you would like to enter a question, go ahead and do that now. We're going to try and get to a couple more here before we end. So the question is CAD detector complementary for non-chromophore compounds?
Obviously, answer is yes, because even though you have RA detector, ELSD detector, CAD is it's a complementary tool for non-chromophoric compounds where you can use for even gradient program also. But again, here one limitation is like sensitivity and linearity. Like you cannot go when you are going for a validation with respect to UV and all. Easily you will get 0.99 as a correlation coefficient. In case of CAD, achieving the 0.99 is not possible. Linearity is a little poor with respect to CAD. But obviously, yes, I will say that it is a complementary tool for non-chromophores where if it is not volatile, you cannot use GC also. In such case, CAD is the preferred thing, but some limitations are there.
Okay, lovely. Here's the next question for you. Are you looking out for a partnership to provide analytical support?
Yeah, obviously, yes, as I cited in my last slide, yeah, we are doing partnership work with many clients analytical method development for the client and method validation, and even for the method transfer also we are doing to the next outsourcing parties, and we do reference standard qualification, impurity isolation and characterization. So we are open for such type of partnership with respect to analytical services.
Okay, thank you very much. And I'm very sorry to everyone who submitted a question we didn't have time for. We are just about out of time now. So we will forward all those questions on, and hopefully you can get a response by email directly. So that just leaves me to thank Dr. V. Srinivasan for what was a great presentation. I'm sure you'll all agree. And also to thank Piramal Pharma Solutions for sponsoring this session. To all the attendees, you will receive an email very shortly, and that will tell you how you can access the on-demand version of this webinar, or you can access it directly through the website, which is www.business-review-webinars.com. Once the webinar ends, today's survey will appear in its place, and we would very much appreciate it if you could answer the questions in there and let us know how we did today. We look forward to sharing further webinars with you. Of course we do, so please keep an eye out on the website that I just mentioned and follow us on Twitter. We are at BR Webinars for daily updates, and join our LinkedIn group as well. That's Business Review Webinars. Dr. Srinivasan, is there anything you'd like to say to the audience right now before we end the webinar?
Yeah, sure. Thank you for giving this opportunity, and thank you for your time to take and attend this webinar. If you have any questions after this thing also, you can write a mail to me. I'll try to answer your questions via mail. And thanks a lot for your time and arranging this seminar, Tarak.
No problem at all. Thank you once again, and I hope everyone in the audience today has a lovely rest of your day. Goodbye.